Welcome to our deep dive into the history of microbiology. Humans have long pondered how life begins. We transition today from ancient philosophical speculation to the cell theory, which states that all life is cellular and that cells only arise from pre-existing cells. Cell theory serves as the foundational pillar of modern biology. It provides a universal framework for understanding how life is structured and reproduced. The theory is built upon three fundamental postulates. First, the cell is defined as the basic functional unit of life. Second, all living organisms, from the simplest bacteria to complex humans, are composed of one or more cells. Third, life exhibits reproductive continuity. This means new cells arise only from the division of pre-existing cells, rather than through spontaneous generation. These three tenets collectively define our biological reality, shifting our perspective from speculation toward empirical cellular science. While all life is cellular, it is not uniform. Prokaryotes and eukaryotes represent the two primary cellular classifications in biology. Prokaryotes are defined by their simplicity. They lack a membrane-bound nucleus, and their genetic material is found in a loose nucleoid region. In contrast, eukaryotes are more complex. They possess a membrane-bound nucleus that protects their DNA and organizes cellular functions into specific organelles. Prokaryotes are typically smaller, while eukaryotes exhibit greater internal organization, allowing for vast metabolic diversity across the tree of life. For millennia, biological thought was dominated by a single belief that life could arise spontaneously from non-living matter. This theory suggested that organisms did not need parents, but could simply emerge from decay or inanimate environments. This phenomenon was often attributed to a mysterious pneuma, or vital life force, thought to be present in the atmosphere or the substance itself. Proponents pointed to common observations, such as frogs seemingly appearing from the mud of the Nile, or insects appearing suddenly on decaying matter, as proof of this spontaneous birth. This remained the dominant scientific and philosophical view of life's origin until the mid-17th century, when experimental rigor began to challenge these long-held assumptions. Aristotle, the influential Greek philosopher, formally articulated the theory of spontaneous generation. He observed life emerging in seemingly empty environments, such as fish appearing in newly formed puddles. He concluded that these non-living materials contained a hidden spirit or pneuma capable of birthing life. This concept dominated biological thought for nearly 2 millennia. This belief, while later proven incorrect, was the accepted scientific wisdom of the ancient world. Throughout the Middle Ages, the theory of spontaneous generation held strong. Observers often witnessed frogs appearing in the Nile River mud following seasonal floods. These were not considered the result of migration, but rather as life created directly from the mud itself. Similarly, the appearance of mice in grain storage was attributed to the transformation of moldy grain into living creatures. This belief persisted for centuries because it matched casual observation, failing to distinguish between spontaneous origin and simple animal movement. In the 17th century, the chemist Jan Baptista van Helmont proposed a surprisingly simple recipe for the spontaneous generation of mice. He claimed that if one placed dirty rags and wheat into an open container, the interaction between these items would spontaneously produce living mice. Van Helmont's error lay in his limited understanding of biological observation. He assumed creation where there was only attraction. The rags and grain were not the source of life, but a resource for mice living in the vicinity. In 1668, Francesco Redi designed a landmark experiment to challenge the theory of spontaneous generation. He placed fresh meat into 3 distinct jars to test the requirement of parental life for the development of maggots. He left one jar open, covered the second with fine gauze, and hermetically sealed the third. Redi observed that flies were attracted to the meat. In the open jar, they landed directly. On the gauze-covered jar, they could only hover above, unable to reach the meat. The result was conclusive. Maggots only appeared in the open jar where flies had direct access. By using these controls, Redi provided the first rigorous scientific evidence that life comes from pre-existing life. This experiment laid the foundation for the scientific method, replacing conjecture with observable, controlled evidence. Redi tested three distinct conditions to verify if life arises spontaneously. He placed meat in open, sealed, and gauze-covered containers. In the open jar, flies entered and laid eggs, resulting in the appearance of maggots. This clearly demonstrated that the maggots were the offspring of the flies. Conversely, in the sealed and gauze-covered jars, the meat remained maggot-free. Because flies were prevented from accessing the meat, no maggots developed. These observations provided the first rigorous experimental evidence against spontaneous generation, supporting the principle of biogenesis, that life only originates from pre-existing life. In 1745, John Needham attempted to revive the theory of spontaneous generation. He hypothesized that a brief boiling of nutrient broth would destroy all existing life. Needham heated the flasks, sealed them with corks, and set them aside. He claimed that the subsequent appearance of microorganisms proved they arose spontaneously. However, his experiment was flawed. The broth was not boiled long enough to kill heat-resistant bacterial spores. Consequently, the microbes were not new life, but survivors of his ineffective sterilization. John Needham's 1745 experiment attempted to prove spontaneous generation by boiling broth in sealed flasks. However, his conclusion relied on a flawed methodology. Needham heated the flasks, but the temperature never reached the critical point needed to kill all microorganisms. Because he didn't boil the broth long enough, heat-resistant microbes survived the process. These survivors continued to reproduce, causing the broth to become cloudy later. This misled Needham into believing they appeared from nothing. His experiment serves as a classic lesson on the vital importance of proper sterilization techniques in biological research. Lazzaro Spallanzani addressed the flaws in previous experiments by introducing more rigorous control. He hypothesized that the microbes observed by John Needham were not arising spontaneously, but were surviving the initial heating process. By boiling the nutrient broth significantly longer and sealing the flasks hermetically, Spallanzani prevented any external microbes from entering. The result was clear. While Needham's broth turned cloudy, indicating microbial life, Spallanzani's sealed and thoroughly boiled broth remained clear indefinitely. This experiment demonstrated that life requires exposure to the environment, providing powerful evidence that spontaneous generation was a misconception. Lazzaro Spallanzani's experiments seemed conclusive. If you boil broth and seal the container, no microbes grow. However, critics like John Needham had a different interpretation. Needham argued that boiling for too long destroyed the essential life force, a mysterious spirit in the air required for life to emerge. By sealing the flask, they claimed Spallanzani had not just sterilized the broth, but had also prevented this vital, life-giving spirit from ever entering the container. In their view, the absence of growth wasn't proof that life comes only from life. Instead, it was evidence that the necessary life force had been excluded. This philosophical challenge highlighted a major problem. How do you distinguish between biological contamination and the presence of a vital spirit? This debate showed that science required more than just sealing a flask. By the 19th century, the scientific debate over whether life could spontaneously arise from non-living matter remained unsettled and deeply polarizing. Two opposing schools of thought locked horns: spontaneous generation and the theory of biogenesis. Despite centuries of experiments, neither side could claim victory. To resolve this, the prestigious Paris Academy of Sciences offered a significant prize for a definitive, rigorous experiment that would finally answer where microbes come from. Louis Pasteur, a brilliant and meticulous French chemist, took up this monumental challenge. He set out to design an experiment that would provide a final verdict on the origin of life. Louis Pasteur sought to settle the debate regarding the role of air in spontaneous generation. He hypothesized that the air itself was not a source of life force, but rather a carrier for microorganisms. Pasteur designed an experiment to filter air through gun cotton. By drawing large volumes of air through this cotton filter, he could capture particles suspended in the atmosphere. When the filter was examined under a microscope, it was clear that it had trapped numerous airborne microorganisms. This provided physical evidence that the air was teeming with biological life. This discovery directly challenged the life force theory. If microbes were present in the air and could be physically trapped, they were not spontaneously arising from nothing, but were instead being introduced from the environment. Pasteur's masterwork was the Swan-Neck flask. Previous experiments were criticized because sealing the flask prevented fresh air, which many believed was essential for life. By drawing the neck of the flask into a long curved shape, Pasteur created a brilliant solution. It allowed air to flow in and out freely, satisfying the critics. Crucially, the complex curve trapped airborne dust and microbes in the bend. The liquid inside remained sterile, despite the open-air connection. This design provided the definitive proof that life does not arise spontaneously, but is introduced from the environment. Pasteur's flask remains an icon of experimental ingenuity. Pasteur boiled the broth in these specially designed flasks to kill any existing life. As long as the neck remained intact, the broth stayed clear indefinitely. The swan-neck design allowed fresh air to enter the flask, satisfying those who claimed air was necessary for life. However, it trapped airborne microbes and dust within the bends of the neck. Because the microbes were trapped, the nutrient-rich broth remained sterile. This was a definitive, controlled demonstration that life requires pre-existing life. Pasteur sought the final proof to debunk spontaneous generation. He had flasks of broth, kept sterile for months due to his swan-neck design, which trapped airborne microbes. To prove that these microbes were indeed in the air, he broke the swan necks of his flasks, exposing the sterile broth directly to the environment. Within only a few days, the once clear broth became cloudy with rapid microbial growth. The experiment confirmed that life does not arise spontaneously. It requires entry of existing life from the air. In 1862, Louis Pasteur's rigorous experiments finally achieved a decisive victory for science, ending centuries of debate. His work cemented the fundamental biological principle, omne vivum ex vivo, which means life only comes from life. With this, the theory of spontaneous generation was officially relegated to the history books, marking the dawn of modern microbiology. This profound discovery shifted the scientific focus toward germ theory and the development of aseptic techniques that save lives today. The history of microbiology is defined by the evolution of scientific rigor. Each experiment from Redi to Pasteur was not just about testing life, but about mastering variables. Scientists learned to isolate environmental factors such as air, heat, and physical access to eliminate bias. By holding these variables constant, they ensured that their results were based on evidence, not assumptions. Today, this standard of control remains the bedrock of medical science. It is the fundamental process used in clinical trials and pharmaceutical validation, ensuring that what we discover today is accurate and reproducible. The refutation of spontaneous generation marked the birth of germ theory. We moved from attributing disease to mysterious life forces to identifying specific microbial agents. This understanding revolutionized medicine. Acknowledging that microbes are living entities led directly to sterile techniques. Surgeons began to wash hands and sterilize instruments, drastically reducing patient infections. Modern hospital safety protocols, which we rely on daily, were established to keep our environments free from contaminating microbes. This rigor is the foundation of modern surgery and pharmaceutical safety. Without this scientific paradigm shift, the high standards of medical care and infection control we expect today would be impossible. Life only comes from life, and our ability to control this principle saves millions of lives every year. Consider the case of Barbara, a 19-year-old presenting with acute respiratory symptoms. In earlier centuries, such illnesses were often attributed to miasma or environmental factors appearing spontaneously. Today, our clinical perspective relies on microbiology. We do not assume spontaneous generation. We search for the specific biological agent responsible for the pathology. By isolating the specific pathogen, clinicians can provide targeted therapy. This methodology is the direct descendant of the work by Redi, Spallanzani, and Pasteur, ensuring that our medical interventions are based on empirical reality. The shift from spontaneous observation to identifying causative agents defines modern healthcare. Barbara's recovery is a testament to the accuracy of this microbiological foundation. From the ancient Pneuma of Aristotle to the rigorous controlled experiments of Louis Pasteur, biology has transformed from speculative myth into a foundational empirical science. Today, our focus has shifted entirely to cellular reproduction and genetic inheritance. These mechanisms are the keys to understanding life, health, and disease in the modern era. The standards of rigor established by these experiments are now the bedrock of clinical trials, vaccine development, and hospital protocols. This journey continues as we apply the scientific method to solve the future's greatest biological challenges.